Hello, welcome to Science Breakthroughs from Science Mission. I'm Sadashiva Pai and going to discuss a couple of breakthrough papers published in the week of June 23rd. Let's start. First, we'll discuss a paper published by Vivek Mahadevan in Cell Reports on the regulation of chloride homeostasis in neurons. Why is this paper so important? Increased intracellular neuronal chloride is linked to numerous neurological disorders including epilepsy, autism, and neuropathic pain. Low intracellular chloride is maintained by KCC2, a neuron-specific potassium chloride cotransporter. Authors elucidate hitherto unknown mechanism of regulation of KCC2. Using whole brain membrane fraction immunoprecipitation experiments, authors demonstrate interaction between KCC2 and glutamate kainate receptor subunits. The interaction is further confirmed by co-immunoprecipitation experiments in HEK293 cells and hippocampal neuronal immunostaining. Authors further demonstrate using antibody shift assay and 2D gel electrophoresis that native KCC2 and glutamate kainate subunits exist in the same oligomeric complex. Authors also show less oligomerization and decreased KCC2 in expression at the surface in glutamate kinase subunit knockout hippocampal neuronal cultures, indicating the importance of glutamate kinase subunits in KCC2 oligomerization and expression. When KCC2 activity was measured by recording the reversal potential for GABA, authors found that eGABA was depolarized in glutamate kainate knockout and shRNA-treated hippocampal neuronal cultures. Thus, this study demonstrates the importance of glutamate kainate receptor subunit for the functioning of KCC2 cotransporter. Next, let's look into a paper published by Colin and colleagues in Nature Neuroscience. Authors try to understand the role of ASIC in addiction. Why is this paper so important? Drug addiction affects millions of people worldwide, but the mechanism is not clearly understood. ASIC role in drug addiction is not known. Authors demonstrate that ACIS through hitherto unknown postsynaptic currents regulates plasticity underlying addiction-related behavior. Deletion of ASIC1A by knockout or ASIC reinjection disrupted acid-evoked currents in the nucleus accumbens region of the mice. Also, ASIC1A knockout and selective partial knockout of ASIC1A in nucleus accumbens mice exhibited a significantly greater cocaine and morphine-conditioned place preference. In addition, ASIC1A injection restored acid-evoked currents and reduced conditioned place preference to cocaine. So, ASIC1A induces acid-evoked currents and required for conditioned place preference. What is the mechanism of action? As shown in this figure, ASIC currents contribute to synaptic transmission. ASIC1A disruption increases dendritic spine density. And as shown here, loss of ASIC1A increases inward rectification of AMPA mediated EPSCs, which is normalized by re-establishing ASIC1A expression. Thus, ASIC plays a role in synaptic transmission, spine density, and glutamate receptor function. Next, authors show that ASIC1A disruption increases AMPA to NMDA ratio, which is acutely reversed by single dose of cocaine. Overexpression of ASIC1A in the rat nucleus accumbens non-significantly enhanced acid-evoked current but significantly enhanced amiloride sensitive synaptic currents, and also attenuated cocaine self-administration. So ASIC1A plays an important role in drug addiction. Thank you for watching Science Mission.